Good afternoon to everyone, and thank you for joining us today. Actually, I'm at the end of my speech, so let's go back to the beginning. Here it is. So I will today present some data on our preclinical candidate for the treatment of seizures in a genetic disorder that is called tuberous sclerosis complex, that we'll refer to TSC for simplicity. So to tell you about TSC, I would like to tell you the story of Mia. Mia is a 4-year-old little girl who actually was diagnosed with TSC prior to birth using ultrasound for heart malformation. This is benign. And at birth, she underwent genetic testing, was confirmed to have TSC, and had MRI, because more than 90% of the patients with TSC actually have brain malformation. She was confirmed to have brain malformation. We also know that the brain malformations lead to seizures, so she had EEG recording as a newborn, and she was found to have a lot of subclinical seizures. By the age of 2 years, Mia unfortunately had one subclinical seizure every 30 minutes. And she went through a barrage of different drug treatment, anti-epileptic drugs. Ultimately, she went to one of the standard of care, which is brain surgery. He helped for a few months, but the seizures came back. She had a second brain surgery, which ultimately did not help. She also has the other standard of care, which is a drug called everolimus, but it's not very effective. She cannot use it at the dosage that would be helpful to treat seizures because of the terrible side effect. So Mia has seizures. She has comorbidities with it, where she will have insomnia and behavioral abnormalities. And this is unfortunately for life.
And let's get started. And once you complete the setup, and I think it's...
Excuse me, someone is talking. Okay, I'll go back. And Mia is actually an example of thousands of patients with TSC in the US. So there is a great need for new drug or any kind of treatment for these patients. And this is our goal. So although TSC is actually an orphan disorder, this is a high cost for patient and for the patients and the society. So there's about 40,000 patients who are drug resistant that have epilepsy need a treatment in the US only. The standard of care everolimus, to give you an idea, costs $16,000 per month per patient. So this is actually an opportunity, a market capital of 5 to 6 billions per year in the US only. So to address this issue, my team has been working on TSC. I'm a tenured professor at Yale. I've published over 100 publications, and we have developed one of the unique model of epilepsy in TSC that is now used by others, scientific lab as well as biotechs. I have connection to a lot of neurologists, as well as the TS Alliance that give us access to a defined population of patients. And we have IP protection through Yale, which I will detail more afterwards. So let me tell you a little more. So as I mentioned, the standard of care, brain surgery and everolimus, is clearly inadequate. However, the everolimus clinical trial can be used to our advantage as it's well-defined. We can use this well-defined clinical endpoints. The primary endpoint for the phase 3 was to look at the % change in seizure frequency over the course of 18 weeks. So it's a highly quantifiable change of seizure activity. So what did we find? We found that a molecule called filamin A is increased in both patients with TSC as well as a different mouse model of TSC, including our mouse model, like I mentioned, is used by others now. So what is filament A? Filament A is actually a fairly large molecule inside cell that is part of the cytoskeleton and has also dozens of binding partners. So what happened in TSC is that in TSC you have mutation in TSC1 or TSC2 that in one hand leads to increase in mTOR activity. mTOR is the target of everolimus. And there's a parallel increase in filament A, which is the target of our small molecule. Together, mTOR and filament A dysregulation will lead to increased cell size, the generation of brain malformation that will lead to seizures. So what I will show you very briefly is that blocking filament A activity or decrease its level shrunk the brain malformation and significantly reduce seizure activity in our mouse model. So we first validated that filament A is actually the proper target by using short hairpin RNA, which is a way to decrease filament A levels. And we found that it decreased this overgrowth of cell size and it significantly reduced seizure activity by 70 to 80% in mice. Then we had access to a small to a small molecule filament A modulator called NT125. And we gave two types of treatment. We studied a neonatal treatment, so very young in these mice at postnatal day 8, which is like a newborn human infant, or in young adult. And in both cases, with these treatments, we were able to significantly reduce seizure activity by about 70%. However, this drug NT125 needs some improvement. The half-life is fairly short, and we would like to have a composition of matter patent. So we are at the phase of lead optimization. We are doing a medicinal chemistry with a CRO NEDP, which is actually part of Genesis, like you saw in the previous talk. We have already synthesized more than 20 compounds. The structure of NT125 is known and published. And we are going through high to medium throughput assays that we have developed in the lab to look both at in vitro efficacy, filament interaction, as well as an in vivo efficacy on cell growth prior to go to seizure activity. So as mentioned, we have IP protection. We have a patent of use for NT125 and any analogs, as well as any method to target filament A to treat epilepsy. Upon the synthesis of some of the drug we have synthesized actually would give us a composition of matter patent. So what about the competition? So as I mentioned, the traditional anti-epileptic drug unfortunately do not work very well. Everolimus has terrible side effect and cannot really be used at high efficacy. And there is a new drug on the block, a new kid on the block, that is called Epidiolex, cannabidiol. Unfortunately, again, the efficacy was only 20% compared to the placebo. And there were, again, severe side effects with this drug. So how does that compare to our drug? In mice, we had 70% efficacy at a very low dose. We used a liquid formulation that can be adapted to children very quickly. And this drug, actually, NT125, is undergoing clinical trial for Alzheimer's disease. Phase 1 and 2 reported no side effect. And we used it at least 10 times less than what they use actually originally in mice for Alzheimer's disease. So in that sense, our drug is a much better profile than the others. Importantly, we can move very quickly from mouse to human. Our study translates very well. We monitor seizure activity, which we can quantify on a daily basis, which will be done in human in the same way. This is the clinical endpoint. So finally, our plan. So lead optimization will be gating to pre-IND meeting. So what have we completed? We have validated our target with shRNA and small molecule. We have ADME prediction already for the new analogs. We have a clinical collaboration in place, an animal model in place, and very clean clinical endpoint for the for clinical trials. So in the next year, where we would be requesting about $2 million to do more medicinal chemistry and validate the candidates, as I mentioned earlier, in vitro, do some BK and BBB permeability analysis for a dozen of candidates, and then test maybe 2 or 3 compounds on seizure activity in collaboration with a CRO who is using our animal model as well as a second model of TSC. Then by 2023, we would work on preclinical formulation with CROs and confirm the BK and BBB to finally have a tox study and then pre-IND application. So this is our plan. And I will finish on that summary slide. Keep it up, and I'm ready to answer your questions.
Terrific. Thank you so much for that presentation, Dr. Bordey. Meghana, would you like to start us off with any questions?
Sure. I just wanted maybe some clarity on the competition slide that you had. Your compound, I was just curious what the preclinical efficacy was for some of the other compounds that are there, and if it was, did kind of tend to show higher efficacy in animals and somewhat a bit of a lower efficacy in once it got into humans. Just wanted to get your comments on the preclinical data for the other assets in the landscape.
It's a great question. It's a bit of a tricky question as well to answer because it's all about dosage.
Mm-hmm.
So if we were to give a little more, we give a very low dose, 10 micromolar to very low dose to 10 to our mice. If we give more, we would have a 100% efficacy, presumably. Everolimus, which is in animal is called rapamycin, has been shown, can show between 40% to 100% efficacy. Again, it's just based on dosage. And most of the studies with everolimus have overdosed by at least tenfold to get a 100% efficacy. We reduced it, and then we had only a 50% efficacy. It was even a higher dose than would be given to the patient. So that much the clinical trial actually that shows that about 50% of the patients show more than 40% efficacy, but they did not detail how much. It was a bit embedded in the clinical trial data. So it's all about dosage, and everolimus just does not get well into the brain.
Thanks.
Great. Christine, do you have a question?
Yeah, that actually answered one of my questions, which is, have you done a head-to-head in your own model to try to understand the efficacy of at least NT125? But maybe along those lines too, as you showed in the mechanism action, which I understand was a very simplified model of that,
Yes.
it does look like there might be overlap in terms of the mechanism ending up on pathways that essentially are at the end of the day doing the same thing in terms of the brain, the cell size. So I'm just wondering if you think this is going to be a combination therapy, if you think in humans you're still going to see 40% efficacy because you're not hitting all of the different pathways that you might need to hit to get better efficacy.
It's a good question, complex. So actually, the increase in mTOR and filamin A are completely independent somehow. And when we block filamin A, we do not touch mTOR pathway. So indeed, that could be used as a combination therapy. It is not our first goal to do that. We would prefer to just do filamin A alone. Eventually down the road, doctors, neurologists will do presumably combination therapy. They all do. The patients have multiple drugs, but our first goal is to do it alone. It's a different pathway. We have identified several mechanisms of action of filamin A downstream to regulate cell size. It is actually one of the mechanisms is presumably by blocking a ribosome biogenesis, for those who know about biology, little molecule that are really important for making more protein. So mTOR increases protein synthesis, the cells get gigantic, And we found that filament A, blocking filament A prevents that at multiple level. It also prevents stress inside the cells through different pathway than mTOR. So we have a little bit of the mechanism in place. But I think we would not go first for combination therapy. Did I answer all your questions, or you have more?
No, I think that's great. Thank you.
Chris, do you have a quick one?
Yeah, sure. Just can you explain the mouse study you did? I know you reduced daily seizures in both the premature and the adult model, but given this the short half-life and the low dose of efficacy, I just better understand what the goals of the optimization program are in terms of, how much longitudinal data do you have on that to sustain that reduction in seizures? Was it multiple dosing per day, or was it once a day? Just explain that mouse
Sure.
model
So
and how
we
that
have-
relates to the goals. Yeah.
Sure. So, for one cohort of the mice, so for all the mice, actually, we gave two dosing per day.
Okay.
And some we studied very young, and we recorded the mice when they are about 2 months of age. So they're adult recording. So we studied at 8 days for 60 days of treatment for the young mice. For the adult cohort, we treated 2 weeks prior to recording for the EEG. So in 2 weeks, we had improvement in seizure activity. And then we did longitudinal studies as well, where we recorded mice they received just saline treatment, and then we injected our drug twice a day. And we could see a progressive decline in seizure activity in these mice in the course of about 2 to 3 weeks. If we were to stop treatment like every other drugs, the seizure will come back.
One quick question. Isn't everolimus now generic as of late 2019? I know generics were approved by the FDA. I just don't know if there's some kind of regulatory exclusivity for the tuberous sclerosis patients.
I think there is, but I do not remember. Yeah, it's a very old drug, obviously used for transplantation, but for the use of TSC, I do not remember the date for the patent, but it's still exclusive.
All right. Thank you very much. Nice presentation.